We'll take a look at Chapter 13 today. Chapter 13 is titled Human Microbe Interactions. One of my favorite chapters, getting into some of the ways that microbes are now being recognized to influence the human body. I wanted to just say before we get into this chapter has changed a lot over the last few years since I've been teaching, changed over the last 10 years with the increase in technology and just further research going into this field, and is going to continue to change even more so over the next 10 to 20 years, and of course further after that. So there's a lot that's changed already. We're going to address that. I just also want to point out there's a lot that we don't know that's going to change over the coming years. So what we say today has to somewhat be taken with a grain of salt, in that it's going to continue to change. And we'll get into that as we go into part one here. So in part one, I'm going to address a lot of these new developments. So one of the interesting things is that we're starting to understand the human body better by actually studying the human microbe interactions. So we've talked about microbes in the gut, we've talked about microbes in the human body in general. And what's interesting is that they're now starting to see the microbes as part of the human body. And so skip down to the bottom real quick. They've actually come up with a new term recently called the holobiont, which describes the human and the microbes, the resident micro, or what's called microbiome, as one kind of entity. Now we can talk about them separately, but there's a new term to represent that these are really kind of a combined thing that you have to really recognize as a unit of multiple parts, as opposed to just the humans and just the bacteria, just the microbes. And that's a recent thing, recent in the last 10 plus years. Scientists have always kind of thought of the human body as one thing and the microbes as another. And now they're realizing that's not really the best way to think about it. If you really want to know how the body works, what makes it tick, you have to recognize that these things actually coincide together. And you can't, for example, just look at the presence or absence of disease as an indicator of the overall status of the microbiota or the microbes in the body. So traditionally, it's always been thought, if you have this bacteria that causes this disease, and therefore we treat this bacteria, we eliminate this disease. And now they're realizing that it's more of the certain bacteria actually affect the way your body functions. That they produce certain enzymes that the body needs. They can even influence your mood and your weight gain, your body type. So you can't think about it as just healthy or sick. You have to think about it almost as an everyday sort of scenario that helps you understand the way your body works in general, as opposed to just what makes you sick. So the relationship is much more complex than anyone could have imagined. And really, they're just starting to uncover what that actually means. And so as I talked about in the beginning there, not only has it changed, but it's going to continue to change. So we have to understand that we only really know a little bit about this, really, compared to what they estimate is yet to be known. So let's start by talking about the microbes themselves. What are these microbes? So we refer often to them as symbiotic microbes, and I've got terms down here we'll get into in a second. But these include the common types that we've talked about. Bacteria, by far the most common and well-studied symbiotic microbes in the human body. The most studied because they're the most common. Archaea, it turns out there's actually archaea in the human body. I've actually not addressed that in previous chapters. In fact, in chapter 4, I talked about these as being kind of extremophiles and only found in rare places in the earth. Turns out that's not exactly accurate. There actually are archaea in the body. Only a few species have been consistently found, but there are at least a couple different strains of archaea that exist in the human gut. Some of these actually produce methane, which is a part of the natural gas. Now, I don't know how much they contribute to our flatulence and our gas production, but there are a couple of methane-producing archaea that have been consistently found in the human body. Don't know much about those. There's not a whole lot of information out there, and what there is very technical, but there's at least a clear indication that there's a few species. Fungi is another one that we tend to find consistently. Most of them are yeast. In fact, I think all of the ones that are consistently found are yeast bacteria, such as Candida species. Candida is one that can sometimes cause a yeast infection, but they're actually part of the normal biota, the symbiotic bacteria, or symbiotic fungi in this case. Viruses, I'm going to talk about this one here a little bit later. Turns out viruses are actually part of the normal biota. They're not necessarily just agents of disease. They are actually present in healthy people in relatively large numbers, and that's something that I am trying to wrap my head around, because as someone who teaches this, I've not actually heard much about anyone talk about this until very, very recently. So in doing a lot of new research for this chapter, I came across this and have much more to kind of figure out there, as I think scientists in general do. Mites, you actually have mites in your eyebrows, believe it or not. Don't try and get them out. They're part of the normal biota. Everyone's got them. There's a couple different species. They are one of the more common sorts of eukaryotic microbes in the human body. And then protozoa, there's actually a couple different types of amoeba, some that live in the mouth and then a couple that live in the gut. Some of these can be a part of dental disease, but for the most part, they're considered normal. Now, there's too much here to get into all of this in detail, so we're talking about this really kind of vaguely. I'm not going to get into all the species just because there's too much to get into, and because a lot of that's still kind of tough to decipher. It's still somewhat new. So, general terms here. Now, there's some common terms that they use to describe the symbiotic microbes. You'll hear all these in different contexts, and our own textbook uses a lot of these actually, and you may hear others that aren't listed here. So things like natural biota, natural flora, those are fairly common, resident biota, and even the human microbiome. They all mean the same thing. They're all talking collectively about these symbiotic microbes. And as I kind of alluded to there, mostly what we know about is the bacteria, and that's because they're the most common. They've been the key focus. The research is ongoing, and they're just going to have to keep doing more and more research. But right now it's been focused on the bacteria. So most of the information is centered around them, and there's fewer sources to really get in-depth information on the other types. And so, as I said earlier, it's changing fast. Every day these studies are emerging. If you follow headline news, even just in the headline science sections, I, for example, follow Google News pretty much daily. And I always scroll down to the science and health sections. And, you know, maybe not every single day, but it's common for a couple articles a week to come out related to this very topic right here. So there's a lot of new things happening. Each study is just one little kind of clue. Very rarely do you have a major study that proves or disproves anything significant, but they all kind of just tell a little story about what's happening. And with all of that in mind, many scientists, microbiologists especially, believe we've kind of entered just recently into a whole new era of microbiology, and that this represents a transition to a new understanding that we really are just kind of walking into. So what's changed? Like, why is it changing? What's going on there? Number one is technology. Like everything, you know, technology is changing the whole world. So no exception here. Specifically, though, that has to do with DNA sequencing. You can now more effectively study living things by studying their DNA directly. It used to be that with bacteria and microbes especially, you had to culture them. You had to grow them on a petri dish or something. And then you had to pluck them off that, put them under a microscope, run tests on them. And while they still do that, it's far less effective because most of these microbes really don't even grow in the lab. And even when they do grow, it's difficult to study them sometimes. Time-consuming, expensive, and limited. So now what they're doing is they're analyzing the DNA. And by sequencing the DNA, you can learn a lot just by understanding the genetic codes themselves. There are limitations to that as well, but it's very cost effective and it's very thorough, and you can study things that you can't actually grow. So you could study many microbes with this mechanism that you could not study traditionally. That, coupled with improvements in computer technology, take the S off that, computer technology is, you know, like a lot of things, that's also helping. So there's a lot of information when it comes to the sequencing, tons and tons and tons of data that has to be stored, and you need really significantly powerful computers to run all that. So that's another big part of that. Coupled with that is an ever-reducing cost in this technology. Real quick on a history of this, if you go back to the early 2000s, it was around 2003 that the very first genome, the human genome really, was sequenced. So one of the first major genome sequencing projects. And they don't even know, from this source I have down here, from the National Human Genome Research Institute, what they were saying was that they don't even know exactly how much it costs. It's difficult apparently to find all the invoices or whatever they're looking at, but they estimated it was somewhere between $500 million and $1 billion. So incredibly expensive. Took them over a decade to do. And by just 3 years or so after the project finished, the cost had dropped down to $20 to $25 million. So a dramatic reduction in the cost within just a couple of years. That's something like a 20 times the reduction in cost within a couple of years. Within 10 years of that, according to this source, the cost had dropped even more, to where in 2016, this is October 4th, 2017. So we're talking this estimate within the last year or so of this recording. But right now, the cost varies between about $1,000 to $1,500, and that cost is just going to keep dropping and dropping and dropping. So it's going to probably end up somewhere in the couple $100 range to sequence. I'm talking about human genomes within probably the next few years, if it's not already close to it now. So we're talking about a dramatic drop. Now, and that's for more complex genomes like the human genome. So bacteria are much cheaper and more cost effective. So a major reduction in the cost is a huge part of that. So technology is the big one, but I think maybe even more important was just an awareness. And they're both important, but awareness just really means that for a long time they didn't understand the significance of the microbiota in the human body. And as they started to recognize the trend that these were showing up in studies indicating their significance, they started to put more and more effort and focus into studying the microbiome. And I think that was probably the biggest impact, really, because the technology is only good if you know how to use it and apply it. So there was a dramatic shift in our understanding that there needed to be a emphasis on studying these. If you look at the emphasis of how microbes were handled between the 1800s and the 1900s, they just basically studied microbes from the sick and the dying for the most part. That's somewhat of an oversimplification, but largely true. They mostly just studied the sick and the dying. They were taking microbes off of the sick and dying and trying to figure out, well, what microbes are causing it, and what can we do with these microbes to stop them, which makes perfect sense, right? I mean, that's the best way to start with the problem, is by taking the obvious and understanding the obvious. And they more or less exhausted all the obvious microbes, and today what they're finding is that to get past that kind of low-hanging fruit, it takes a lot more work and a lot more awareness and dedication. So this period between the 18 and 1900s was referred to as the golden age of microbiology. And as I mentioned before, we're kind of entering a new era of microbiology. I'm not sure if anybody's coined a new phrase for it, but that's the idea in general there. So although the discoveries that they made during this time period, the 18 to 1900s, it turns out they were just the tip of the iceberg. Again, kind of like the low-hanging fruit. They didn't realize at the time that the vast majority of microbes could not be cultured, so they were doing a lot of culturing to analyze. They didn't realize that the quote-unquote normal biota might even cause disease. It was always thought that invading microbes from the outside world, coming into your body, like an invading robber or something, were the ones that were making you sick. They now realize it's actually sometimes a shift in the balance of your quote-unquote normal biota that can actually cause disease. And I don't think anyone really appreciated at all at that time that microbes could actually be responsible for more subtle conditions such as depression, anxiety, and obesity. They were always thought and recognized to be contributors of tuberculosis and staph infections and all sorts of fairly serious types of infections, but malaria. But now they're realizing there's a more subtle nature to the effects that they have. So once that they understand this, now they're setting out to kind of figure that out in more detail. So technology and awareness are some of the two key things that are changing this whole field. And with that in mind, it's going to take a long time. They've been at it pretty heavily for about 10 years now. Some of the most significant studies that we'll talk about next started in around 2007, and they got a long way to go. So most of the microbiologists are referring to microbiology and the study of the human microbiome as being in its infantile stages. Kind of gives you an idea of where they're at. They're learning to crawl, so to speak. And with that in mind, they're going to get stuff wrong. There's going to be some claims that are being made today that are either entirely wrong, understated, exactly right, or anywhere in between. So again, I mentioned that in the beginning, kind of take everything with a grain of salt and understand that it's based on what we know today, and that might change tomorrow. As an example of something that's already been overturned, and a long-stated belief is that the ratio of human cells to microbial cells is 10 to 1. Turns out that's not true. This is something that up until just recently, I think up until the last version of our textbook, actually, this was stated. And there was apparently a study that was done, I think in the 1970s or 80s, from a scientist who came up with this estimate, and it was published, and it was just passed along ever since. And it turns out it's entirely wrong, according to the latest research. Now, maybe this research is wrong, but based on the latest research, it appears that ratio is not only wrong, but it was way off. It wasn't even close. So it's interesting not only that they were wrong here, but how easily bad information is passed along in the scientific world. It's an example of where sometimes you think there's a lot of thorough research that goes on in order for a fact to be passed around like it is. You find out sometimes that nope, there's not the research really to back it. It was stated incorrectly, and it kind of caught like wildfire, so to speak, and false information gets passed around. And so this is one of them. So the ratio, rather than being 10 to 1, turns out to be closer to 1 to 1. So they were saying in the beginning that for every 1 human cell you had, there were 10 microbial cells. And it turns out that ratio is about a 1 to 1, which by the way is still fairly significant. Like the fact that you have 1 microbial cell in and on your body for every 1 of your own cells, that's a pretty astonishing thing. The fact that you're basically, quantitatively speaking, equal microbe to human cells. Now our cells are bigger and they take up significantly more mass, so there's no, that ratio is not even close in terms of the mass or the volume, but in terms of quantity, then it's about a 1 to 1 ratio. It does vary, by the way, and they said it may be closer to 1.2 to 1.3, but certainly closer to a 1 to 1 than it is a 10 to 1. A lot of the information that's changing is at the result of what's called the Human Microbiome Project. The Human Microbiome Project was one of the most, and still is one of the most major studies that is kind of driving forward this information in the field. Started in 2007, going on for about 10 years now. Their major goal is to do the genome DNA sequencing that I mentioned earlier, using some of the big data tools, the supercomputers, in order to store and process all this information. So the effort has been largely focused on genome and DNA sequencing. So their goals and objectives are to collect these sequences. From there, though, they're trying to determine what microbes are present on the human body. And it's different for every single person. So that's a very difficult task. So they're not only having to just study microbes in general, but trying to compare from person to person. And they're collecting large databases that kind of analyze all the different strains and how they vary, trying to get an idea of the quantity, not just naming the species, but how many and what are the proportions, how those proportions change from person to person. Fairly huge effort, really. And again, that focuses largely on the unculturable microbes, because the culturable microbes up until this time had been studied, because that's again how they studied things traditionally, was to culture and then study. And now they're looking at largely the unculturable, which make up the majority. And then the biggest, most difficult is to determine the role that these microbes play. And I believe that that's going to just be an ever ongoing study. I don't see it ever concluding, certainly in my lifetime. Maybe I'm wrong about that, but I think that this is the million-dollar question. And it's one thing to say what's there, and even in the proportions, which is difficult, is certainly more obtainable. But to then go on to say what role, what are they exactly doing, I think it's obtainable, but I think it's way more difficult. So I think that's going to be the most difficult of these objectives to actually complete. But with that in mind, they are already making progress. There's already some fairly interesting things that have come out of it. So real quick, let's take a look at some of the results that have been published so far. Some of them, this is not all of them, by the way, this is some of the most notable ones here. Starting off, there is a recognition that the genetic contribution from microbes is significantly larger than our own human genetic contribution. So if you look at the amount of protein coding genes in the human body, there are approximately 22,000. In fact, that number that actually may be higher. I've seen other studies that put it closer to 19 to 20,000. They actually don't know exactly. It's apparently not that easy to say exactly how many you have, but it might be closer to 20,000. But it's not that many in comparison to what compared to, if you look at the microbes here, the combined contribution of the microbiome, of which there are thousands of different species, could be as high as 8 million of these protein coding genes. So real quick, just want to reiterate this in your head real quick. Remember the central dogma of biology, which is that DNA produces RNA, which produces protein. And then I've said that the protein does the quote-unquote work of the cell, right? Makes the cell tick, also makes up the structures of the cell. But in this case, thinking of work in all different contexts, your body makes approximately 22,000 of these different workers, so to speak, or worker tools that we talked about in chapter 7 and 8, of these kind of being tools. Microbes produce 8 million of them. Okay, now they are largely confined to the intestine. Some of these are in the skin, some of these are in the mouth, some of these are in the lungs. They're all different parts of the body. But the contribution of different proteins in the body from microbes to humans is significantly different. This is over 300 times more, about 365 times. And what they're starting to realize is that your body not only requires the proteins that your cells make, but it requires some combination of the proteins that these microbes make. And exactly what those are and exactly the implications is going to be part of the understanding the health and disease, as I mentioned, the more difficult part of the picture. But the recognition that there's a dramatic difference in the number is a big kind of step into the unknown there, and it's just recognizing that there's a huge area of potential here in understanding what these do and how those contribute. We know that when microbes get eliminated from the body, certain conditions tend to pop up, things like immune disorders and obesity, and even in neurological disorders. And so there's a lot of speculation already that these proteins may be part of those disorders, or the lack of those proteins. And just one more thing, this is only just an average. Each person can be different. You may have half that many, you may have more than that. So that's another factor that may differ from person to person. How many different protein encoding genes do these microbes actually make in your body? Next thing is that when they were studying the individuals, they found that there were previously sites in your body that were thought to be sterile. For example, they thought the lungs were naturally sterile and that you only had microbes there during infection. That turns out not to be true. They're finding that with the blood as well. A lot of people thought the blood was relatively sterile. Now they're finding small trace amounts of microbes there. So the idea that you have sterile locations in the body turns out to appear apparently here not to be true. This one I found really interesting. All of the healthy people studied in the Human Microbiome Project, all of them had dangerous pathogens, so species of microbe that we would consider pathogenic, in low numbers. And what that means is that you have the potential to have an infection arise from within, not just as an infiltration from the outside world. And I'll talk about that a little bit later as well. But the idea is that sometimes it's an imbalance of microbes rather than an infection that actually causes a disorder, oftentimes more subtle. This one here I mentioned earlier, vast quantities of viruses are found in healthy individuals. This one really kind of is hard to wrap my head around. We tend to think of viruses, as I've even taught in chapter 6, and I have to go back and really revise all of this and have to do some more research here. But they're finding that as a healthy individual, you do have viruses that are floating around your body and don't appear to be causing infection and disease. They're part of the natural status of things. What exactly they're doing is much more difficult to say, and I certainly am not educated enough on this to speak about it, and I think very few people are, because this is a relatively new a relatively new thing, is the fact that you have all these viruses as a healthy individual. So very fascinating there. And then gut bacteria have subtle effects or can influence your overall health. That's another big result. So they've been talking about obesity for a long time. That's now become fairly well understood, at least in terms of the gut bacteria that you have will affect your body type. Certain strains are now being recognized to contribute to higher rates of obesity. Certain strains are recognized to contribute to a more lean body type. So that's more of a long-term understanding in terms of this. So they're stating that fairly confidently now. Another one that just recently became much more understood is Crohn's disease. Crohn's disease is an issue in the intestines, condition that can cause inflammation, and is now recognized to be caused by certain species of bacteria. And if I remember correctly, they found that there was actually 2 species of bacteria, one of them being E. coli, in combination with a certain strain of fungi. So there was a certain group of microbes that came together, and their combination led to a biofilm formation that would trigger inflammation. I'm not sure if it's always the same 3 species, but the study I read indicated that there were 3 known species that were contributing in the individual studied. So it's probably more complex than that. But Crohn's disease is now being recognized to be caused by certain combinations of microbes in the gut, which is good because the assumption there is that they'll figure out a way to treat that. Now, lots of other preliminary research is going on. So in these cases down here, there's not as much definitive evidence, but lots of speculation that will either need further research to either confirm or deny these claims. But heart disease, asthma, autism even, diabetes, and mood disorders are all now being commonly recognized as likely microbes that are likely contributing to these. Now, these are all relatively complex as well, so I don't think anybody's stating that these are always caused by microbes. I don't think most people are stating that, but that microbes can contribute to them in some cases. It may turn out that they're the sole contributors. It may turn out that microbes are what caused them, but I don't think anybody's saying that yet. I think what they're saying is that there are lots of scenarios where microbes may be contributing, or at least partly contributing. Okay, so that's a quick overview of some of the newest things coming out of this area. This next part here goes into more of what's been known about the microbiome. More of this comes right out of our textbook here. So, for example, acquiring normal biota. It's recognized that we pick up certain microbes over our lifetime. But most microbes that we come in contact with are removed or destroyed by our host defense system. And that's before they have a chance to what's called establish. Establishment is kind of the technical word for being colonized in the human body. We'll talk about that more in part 2. So most of the time, microbes we come in contact with, they're destroyed. A small percent are able to establish, but so in other words, they're kind of able to colonize. But in that process, they ultimately attract our immune system that comes in and eliminates them. So for the small percent that do establish, they get eliminated. For even a smaller percent, what ultimately become our normal biota or the microbiome, they are able to somehow establish without drawing attention from the immune system. How exactly that works is still a big part of the ongoing research. But it's recognized that there are these kind of special microbes, so to speak, that your body more or less tolerates, and they are allowed to persist, whereas other microbes are not. And that's certainly not doing it the full justice there, but that is a quick idea of kind of acquiring. So your body is tailored to eliminate and select for certain strains. And we'll talk a little bit more about how that takes place early in your development. These microbes ultimately go on to establish the mutualistic and commensal relationships we've talked about in previous chapters, beneficial or neutral type relationships. They found that your normal biota varies very significantly from person to person, and even from location to location within the body of a single person. So for example, your left hand may have different bacteria, a significantly different array of bacteria than your right hand. That was pretty consistent in all the individuals studied in this project. certainly varies from person to person, but what was really interesting was location to location. So apparently what you do with each hand, typically you have a dominant hand. Apparently, shaking other people's hands, touching door handles, opening, closing, etc., that apparently exposes one hand to a different array of microbes compared to the other, and that ultimately leads to a different composition of bacteria. So it's not only different from person to person, but location to location. What was interesting, though, was that it turns out that although the exact species are not consistent, what is consistent are the quote-unquote protein profiles that they produce. So just going back here real quick, remember it's a lot about these proteins here, these 8 million proteins that these microbes are producing. It turns out that even though the species are different, the proteins are actually relatively consistent. And apparently that's why you can have so much diversity of species without having any real effect on the body, because ultimately the products that they're making are basically the same. And so it doesn't matter so much what you call the species as so much as it does the types of products that the microbes themselves make in the body. So even though the species vary, the profiles remain consistent. So I thought that was kind of interesting. What's been known for some time is that the normal biota benefit the human body in one real obvious way, which is called microbial antagonism. And there's other ways to describe it. I've heard it sometimes referred to as colonial inhibition, where the idea is that your normal biota basically kind of ward off invading microbes. They tend to have an inhibitory or antagonistic effect on these quote-unquote invading microbes. And this generally works in two ways. One is that they occupy space in the body and basically just block out other microbes from attaching. There's only so many seats, so to speak, that can be taken, and your microbes occupy all those seats, and there's nowhere for these other microbes to come in and sit down. At the same time, they also ward them off through releasing chemicals that would otherwise be harmful to these invading microbes. So chemicals that they can withstand that are noxious or harmful to other microbes. So it's kind of a combination of those two, and that combination makes it difficult for outside microbes to just kind of wander into the body and establish very easily. Now, so that's a fairly well-known effect that the microbiota have. Other things in speculation are that they influence the development of organs. It's actually fairly well known that they do have an effect on that. I think it's more difficult to say why. Maybe I'm just not as educated on that, but I know I've seen lots of studies talking about that they do affect the development. I'm not sure they know the mechanism there. We know that they aid in the digestion of food, and that they even can provide key nutrients in the body, such as vitamin K, is one that's been known about for a long time. Microbes produce vitamin B12, but usually they produce it lower in the colon, where we don't absorb it in the small intestine. So they provide key nutrients, some of those which are readily available, some of which are not. So those are some of the benefits they have. Now, as a counterpoint to this, and sort of a side note, your normal biota, what we consider the good bacteria, you have to almost consider them good in the right context. And the context depends largely on the number and the balance of species. So then it turns out that the normal biota may actually be harmful in large numbers, or with really an imbalance of the different strains or species. And they found that when something like an antibiotic is taken, that can really disrupt that balance, and it can lead to what would be considered normal biota taking over the body in a harmful way. The most obvious example of this is the bacteria C. diff, or Clostridium difficile. It's a microbe that's actually somewhat commonly found in the guts of humans. Not always. It's not a staple of the human gut, but it's not uncommon for people to be studied and find this in their gut, that people that are totally healthy. And what's happening here, this is again fairly well understood with C. diff, is that typically an antibiotic regimen wipes out the normal balance of the gut bacteria and leaves the C. diff, typically unaffected by the antibiotic, leaves the C. diff in the gut with more room and space to grow, with the lack of competition from the other microbes that were eliminated. And as it grows into larger numbers, its presence becomes harmful. So when it's in a low dose or low numbers, it's not a big deal. But when it gets to be really numerous, then that particular species becomes really problematic. So that's one of the more obvious examples of an imbalance caused by something like an antibiotic. Diet might be a part of that, probably not with C. diff so much, but more subtle effects might come with diet and lifestyle choices. Okay, one of the last things here I want to talk about with part one was the question here, when does it start? And really it comes down to the first day that you're born, and probably going even before that in your development in utero. So this one is starting to become more clear, but for a long time it was considered that you were sterile in the placenta during your embryonic development. I'm kind of quoting one study here, and so maybe there's other studies to contradict this, but it turns out that that's probably not the case, that you're probably not sterile. This study here in particular pointed to the fact that they found several strains of microbe in what's called the meconium, which is the first stool produced by an infant. They found several different species in the infant gut, which is a pretty clear indication that you're not actually sterile. There are microbes, at least in the gut of developing infants, which it's really not that surprising to me. It doesn't appear that there's a lot of them. It appears that the diversity was pretty scarce, and that there's a lot more to come immediately following your actual birth or the parturition. But it turns out that it's likely in utero you're actually first inoculated. Probably has a lot to do with what the mother's eating. What is definitely known, and what's been well known for quite a while, is that the day you're born is one of the biggest and most significant colonization events to occur in your lifetime. So the moment you're born, you come out of the womb and you're exposed to the world of microbes. And that's whether you have some colonization in the gut or not, this is a major, major, major event. Because even if you have, based on this research, they're showing there's some, but not very many. So most of the bacteria that you pick up are going to come the moment you're born. Now, this leads to another interesting scenario, which is based on the mechanism by which you're born. So as you may probably already aware, you can be born from a more natural vaginal birth or the more artificial cesarean section or cesarean birth. And it turns out that the mechanism matters significantly in terms of the types of microbes that you pick up. So a vaginal birth will expose you to certain strains of bacteria, like bifidobacterium is one, and the cesarean birth will expose you to different types of bacteria, especially those that come from the skin, not only from the mother, but from close relatives, doctors, hospital staff, anybody the baby comes in contact with within the first couple of days. And based on the preliminary studies, it appears that matters. It's difficult to say because there's probably a lot of variability there. This is one of those areas where there's quite a bit of research, but there's going to need to be a lot more to make more clear statements here. But from the research that I've seen, they're starting to become more confident that this is pretty significant and that the difference will matter, not only in the short term, but potentially over the entire lifetime of the child. So here's talking about the consequences here. What they're finding is that the exposure to microbes, even within the first few days, sets the stage, so to speak, for the development of your immune system. And what they're starting to realize is that, let me just go back here real quick to point where I mentioned this earlier, why the body tolerates a small percent of normal biota. Remember I said here most microbes are removed, some established, they're typipally removed. Only a small percent actually stick in the body and stay without being killed off by the immune system. What appears to be happening is that the microbes that you're exposed to in the first few days of your life set the stage for kind of what the body ultimately tolerates later. So the very first species kind of set the immune system to tolerate them. And then later, similar species that come along can be tolerated as well. And so if you're exposed to different strains, the way your body gauges tolerance can be shifted very early on. and it doesn't seem like you can change that very easily thereafter. So taking, probiotics or eating different food may not really have the effect that you're looking for in order to shift that back later. So as it looks as of right now, it matters what happens in the first few days. It sets your immune system up. Not that you're going to die one way or the other, but you might be more susceptible to conditions we'll talk about here in a second. So immune tolerance, vaginal births appear to correctly shape the immune system, for lack of a better description there, where C-sections may more or less do the opposite. Now, this is highly variable. There's lots of things happening. Skin bacteria versus vaginal bacteria is just one of the thousands of variables that happen in the first few days of your life and the first few years of your life. So it's one of the reasons why it's so difficult, because there's so many variables. It's hard to say whether a 4-year-old has a certain condition because of the way that they were born, or because of the food they've eaten, or because of their genetics, or because of the TV they've been watching. So it's really hard to say, but it appears that the mechanism of birth is a significant contribution to their overall health. Back up here, I skipped over this here. The diseases that they're pointing to that could be coming from the C-section are things like asthma is a big one. That's the one I've been hearing about the longest, but obesity is another one. And now I'm hearing more about eczema and diabetes, and then others that are not being talked about as much. But these 4 here are becoming some of the more common discussed in this here, asthma and obesity especially. And I pulled this from a doctor named Dr. Mark Sloan, who's a pediatrician. He wrote a book called Birthday, A Pediatrician Explores the Science, the History, and the Wonder of Childbirth. And I didn't actually read the book, to be fair. I pulled this off of a kind of a summary of this particular area of the book, talking about the gut bacteria. But what he was saying in reference to this part of the book is that the cesarean section may sometimes be necessary, but it should be something that should be avoided, and that if it is necessary for the C-section, that you should try to do something to normalize the gut microbiota of a cesarean-born baby. So you need to understand that it makes a difference, and that even though it's not ideal to have the C-section, there are some things you can do to hopefully kind of counter that. He talked about probiotics as one, although the research so far has not been optimistic on that. So probiotics, as you may know, are just pills and supplements that have good bacteria in them that you swallow like any other pill and try and recolonize the gut that way. And it hasn't so far shown a lot of promise. This one here, there's not a lot of studies as of yet to point to that I've seen anyway. Maybe there are that I haven't seen, but the idea is that you can simply just wipe the baby down with some of the maternal secretions and try and simulate a vaginal birth like you would if the baby had passed through the vaginal canal. That seems to be, to me, to be the most common sense, is to actually just kind of artificially recreate that process. That may be more difficult than it sounds, but I think it certainly would be possible, and something that doctors should be trying, given the implications for how significant this can be. And this last one down here, fecal transplant, would be transferring some of the fecal bacteria into the baby like they would do with certain patients today. This is becoming a more common way of treating gut disorders and C. diff infections. Problem is, with a small child, this could be highly dangerous. I don't think they've done a lot of it. I think this is mostly just theoretical. I don't think they're doing it because there's a high risk there. A little baby doesn't have much of an immune system, and if you transfer the wrong kind of gut bacteria from an adult, that could be catastrophic at a young age like that. So that seems highly risky, although possible. I'm not sure how you study that without putting babies at high risk, but again, I could be wrong about that. So anyway, those are some of the mechanisms that he was proposing there. Okay, so that's the end of part 1 here. And in part 2, we're going to take a look at the process of infection and some of the things that are happening when microbes start to become pathogenic in the body. So take a more look at the more traditional aspect of the negative human microbe interactions. So I'll see you in part 2.